Your drug candidate just performed brilliantly in your lab. The chemistry looks right, the animal data looks right, but before it ever reaches a human patient, before a single IND is filed or a pivotal trial starts, another lab, one you may never visit, is going to measure what your drug actually does inside a living body. And regulators are going to scrutinize every number that lab produces. One gap in that data package, one failed audit, and your approval timeline shifts by more than a year. Hello, I'm Raghuveer Singh Parmar, Director Scientific Affairs at ResolveMass Laboratories, and I work with pharma and biotech teams every day to design and execute bioanalytical programs that hold up under regulatory review. Today, we are going to pull back the curtain on how bioanalytical services actually work, what they cover, why the platform choice matters, and how teams in North America navigate FDA, Health Canada, and new ICH M10 standard. Here is something that often surprises early stage teams. Bioanalytical work begins before you even file your IND. Method development starts at the preclinical stage. You need to know you can measure your drug in plasma before you put it into your human. From phase 1's first in human PK profiles through phase 2 and 3's pivotal studies and to the final data package the FDA or Health Canada will review. Bioanalytical data is the thread that runs through all of it. Every PK parameter, your AUC, your Cmax, your half-life, is the only as reliable as the method that produced it. So what do bioanalytical services actually include? At the core, it is the validated measurement of the drug concentrations and biomarkers in biological matrices, plasma, blood, urine, cerebrospinal fluid, tissue. PK and PD studies give you the concentration time profile that defines dosing. Immunogenicity testing, anti-drug antibody and neutralizing antibody detection is mandatory for every biologic. Biomarker quantitation supports both efficacy and safety endpoints. And undergirding all of it is the method validation, the rigorous documented proof that your measurement system does what you claim it does and the conditions it will be used. Small molecule versus large molecule. So this is where teams sometimes underestimate the complexity. Small molecule and large molecule programs live in genuinely different technical worlds. For small molecules, LC-MS has become the gold standard. It gives you specificity, sensitivity in the program picogram per milliliter range, and throughout you need for large clinical sample sets. For biologics, you are in the ligand binding assay space. ELISA, electrochemiluminescence platforms like MST, or increasingly hybrid LBA, LC-MS for protein quantitation, where immunocapture alone isn't sufficient. Each platform has its own validation requirements, its own failure modes, its own quirks around matrix effects and endogenous interference. For teams operating across North America and globally, you are dealing with at least 2 regulatory agencies and often 3. FDA's 2018 Bioanalytical Method Validation Guidance sets the bar in the United States. EMA has its own BMV guideline which subtly different acceptance criteria, and if you are pursuing global approval, both matter. Health Canada has aligned increasingly with ICH M10, the first globally harmonized bioanalytical validation standard, published in 2022. Knowing which guideline governs your specific study type and understanding where they diverge is not academic. It is the difference between a clean package review and a deficiency letter. Not every bioanalytical study demands the same level of validation rigor, but knowing which tier applies to your study is non-negotiable. Full validation is required for any GLP regulated study that will go into a pivotal submission. Partial validation covers minor modifications, changing from one instrument to another in the same lab, for instance, or adding a new metabolite to an existing method. Cross-validation becomes critical when you change labs mid-study or use multiple methods for the same analyte. At ResolveMass, we have seen sponsors come in with partial validation packages for studies that FDA squarely classified as pivotal. That conversation with the regulator is never a pleasant one. The cases where bioanalytical data fails are instructive. Biosimilar programs have received FDA complete response letters, specifically citing inadequate immunogenicity assay platforms, ADA assay sensitivity below the required drug tolerance threshold, cut point calculation errors. Generic ANDA programs have failed PK bridging studies because matrix effects weren't properly characterized during validation, leading to assay bias that only become apparent in the real sample set. Oncology programs have seen phase 3 data challenge when biomarker assays lacked fit for purpose validation documentation. The pattern is consistent. The shortcut taken during method development or validation is the one that surfaces at the worst possible moment. So what does a well-run bioanalytical program actually look like from the inside? It starts at study design, choosing your matrix, your analytes, your collection time points, and your assay platform before samples are ever collected. That is the step most teams shortchange. Method development follows iterative optimization of chromatographic conditions, extraction efficiency, ionization, calibration range, then full validation, the formal documented proof of accuracy, precision, selectivity, linearity, stability, and matrix effects across the specific conditions of your study. Sample analysis runs against a validated method with in-study QC samples that confirm performance continues to hold. And then a rigorous data review and report with a complete audit trail that shows every integration, every reinjection, every decision made along the way. Regulators review bioanalytical data not just for the numbers, but for the integrity of the process that produced them. So what should you walk away with this discussion? Bioanalytical data is the foundation of every regulatory filing. It cannot be an afterthought. Platform choice depends on your molecule type, and two worlds have genuinely different requirements. FDA, EMA, and Health Canada each have distinct expectations, and ICH M10 is now the global harmonization benchmark you need to know. And design the program early, fixing the validation gap before first collection cost a fraction of what it cost after. If you are working on an IND, an ANDA, or biologics programs and need a bioanalytical CRO partner who understands both the science and the regulatory expectations in North America, ResolveMass Laboratories supports small and mid-sized pharma and biotech teams from method development all the way through the final data reporting. Thank you so much for watching this session and see you in the next one. Until then, goodbye for now.